Good morning, everyone. My name is Kathya Alonzo, and today's topic is going to be about activating sting in vascular and pulmonary syndrome. Definition: Pulmonary vascular disease is a broad term including any condition that affects the blood vessels within the lungs. These vessels take blood that is depleted of oxygen to the lungs from the right side of the heart, and the deoxygenated blood travels through the pulmonary arteries where oxygen is taken up. In the case of this article, they analyzed the DNA of an index patient with an early onset systemic inflammation, cutaneous vasculopathy, and pulmonary inflammation.
The subject of this lecture are pulmonary vascular diseases. Pulmonary circulation problems are very important. In particular, pulmonary emboli and coronary are both very common conditions which many patients will present with and you need to know about. Less common problems are pulmonary hypertension, which are independent of pulmonary, i.e. not associated with chronic hypoxic lung disease, rare inflammatory disease of the pulmonary circulation called pulmonary vasculitis, and arteriovenous malformations, which is where pulmonary artery, is directly connected to a pulmonary vein, and this is a congenital abnormality, which is very rare, and I'm not going to discuss in any more detail today. Major hemoptysis, which might be considered a problem of the pulmonary circulation, is discussed in the lecture on respiratory airways disease. Some general principles about pulmonary circulation. The first is that the symptoms are not specific when you have pulmonary artery problems. That makes it hard to diagnose or think about the condition clinically. There are very few signs, which makes it even harder to identify clinically. And the chest X-ray is actually usually not particularly helpful, again making the diagnosis difficult. Finally, lung diseases in general would normally affect spirometry, but pulmonary vascular diseases are the exception. They do not affect lung volumes or the spirometry values, the FEV1 or the FVC. They do affect the transfer factor. So when requesting lung function tests and thinking about pulmonary vascular disease, you need a transfer factor to be able to identify whether they're present or not. So, diagnosis of pulmonary circulation problems requires some form of imaging of the pulmonary arteries. That's normally a CT scan, but could be an MRI scan, or could even be invasive angiography. It requires some form of imaging of the consequences of pulmonary artery circulation problems, and that is usually an echocardiogram to look at the right ventricle and the pulmonary artery pressure. And these tests are not routinely done on every patient. And that means you need to have a high index of suspicion and order these tests when you think there may be a pulmonary vascular problem.
Application in medicine. With a sting-ligand cyclic guanosine monophosphate adenosine monophosphate, we stimulated peripheral blood mononuclear cells and fibroblasts from patients and controls, as well as commercially obtained endothelial cells, and then assayed transcription of IFMB1, the gene encoding interferon beta, in the stimulated cells.
My name is Manfred Böhm. I'm a senior investigator at the National Heart, Lung and Blood Institute. We are interested in rare inherited vascular diseases in children. We use next-gen sequencing to identify the gene that causes stroke in children. By using this information, we then created a model that explains how the genetic change is linking to the clinical phenotype. This is called in vitro disease model. So we understand the process that leads from the genetic alteration to the disease. And because we understand that, we were able to use drugs and medication that interferes with these pathway and helps the patient to become better, to don't suffer from more strokes. So I'm basically looking for patients who have very unusual blood vessels, and because they are so unusual, they in many cases are caused by genetic changes in their genome that's inherited through their families. That gives us a tool to actually explore them in very detail. We are working to find more answers on ADA2, so these are the gene that's affected. because we only understand the tip of the iceberg, what is actually happening there. We can treat it, but we don't know how long the children will be disease-free. So we need to invest more to fully understand how this change in this one protein affects these children. We can also use the enormous resources of basic science that we have here at the NIH and find out what is the underlying cause for this disease. The fact that what we are finding here at the NIH can actually be used by our outside colleagues to treat these patients with rare conditions and maybe use this information also to treat other patients with more common forms. This is what is all about what we are doing here. So this is our ultimate goal, to generate answers to questions that can be used by our colleagues to help the American people.
Pros and cons. The pros are that with this knowledge, blocking sting may therefore represent an experimental therapeutic strategy in interferon-mediated diseases other than SAVI. Also, options for treating patients with SAVI are currently limited, but the study suggests that the blockade of interferon signaling with a JAK inhibitor may offer a therapeutic strategy. But the cons are that STING pathway can activate endothelial cells directly and induce upregulation of interferon response genes, apoptosis pathway genes, and tissue factor expression, which is a potent initiator of coagulation cascade. Also, the clinical response to interleukin-1 inhibition and to interleukin-1 inhibiting therapy was ineffective in one of the patients.
Tumors grow unchecked by evading the immune system, even though immune cells are present in the tumor and its surroundings. An agonist targeting the protein called stimulator of interferon genes, or STING, is one strategy that can be used to kickstart the immune system into action. The STING agonist is delivered directly into the tumor and works by activating the C-gas STING DNA sensing pathway that is normally activated when DNA is detected outside of the cell nucleus. For example, during the event of a viral infection, initiating an immune response. The pathway induces target gene activation and triggers type 1 interferon secretion. Type 1 interferon activates immune cells, including dendritic cells that migrate to the draining lymph node, where they present antigens, leading to priming, education, and activation of cytotoxic T cells. Activated T cells are now able to find, infiltrate, and destroy the tumor cells. However, tumors can often disarm the activated cytotoxic T cells by expressing proteins such as PD-L1 on their surface, which binds to the PD-1 receptor on T cells and suppresses their tumor-killing functions. By combining the STING agonist with a PD-1 inhibitor, the cytotoxic T cells can remain active, and greater anti-tumor activity may be achieved.
Issue applied in Ecuador. In Ecuador, there are no statistical data of patients with pulmonary hypertension. However, this disease is considered catastrophic, and there are 14 drugs approved in the world for this pathology. Unfortunately, in Ecuador, there are only 2, which limits the improvement of these patients.
Hi, I'm scientist Cindy. Let's learn about the circulatory system. The 3 components of the circulatory system are the heart, the blood, and the blood vessels. The circulatory system, also called the cardiovascular system, functions to circulate blood through the body. The cardiovascular system has red blood cells. Red blood cells carry oxygen to the tissues of the body. The red blood cells also carry away carbon dioxide from the cells. The function of the heart is to pump blood around the body. The heart does this by creating a pressure gradient. that forces the blood to move through the blood vessels. The function of the blood vessels is to provide the network of passageways that allows the blood to travel throughout the body. The circulatory system circulates blood around two loops. The first circuit is the pulmonary circuit. The pulmonary circuit brings blood from the heart to the lungs and back to the heart. The second system is called the systemic circuit. The systemic circuit brings blood from the heart to the rest of the body and back to the heart. There are two major types of blood vessels, arteries and veins. Arteries carry blood away from the heart. Veins carry blood toward the heart. Arteries are usually depicted as red, and veins are usually depicted as blue on anatomical models and illustrations. But be careful, because this is not always the case. The red coloring in illustrations and anatomical models that the blood carried in those blood vessels is oxygenated. The blue coloring in illustrations and anatomical models indicates that the blood carried in those blood vessels is deoxygenated. In the pulmonary circuit, the heart pumps deoxygenated blood to the lungs. These blood vessels are going away from the heart, so they are called arteries. These are pulmonary arteries, but they are always colored blue because the blood they carry is deoxygenated. The red blood cells travel to the lungs where they release carbon dioxide and pick up oxygen. Now that the red blood cells are oxygenated, they return to the heart to get another push before traveling through the body in the systemic circuit. These blood vessels are headed toward the heart, so they are veins. But these veins are colored red because the blood they carry is oxygenated. This newly oxygenated blood travels back to the heart through the pulmonary veins. Here in the heart, the blood gets a final push before its long trek around the body.
Conclusion. The study of autoinflammatory diseases has uncovered mechanisms underlying cytokine dysregulation and inflammation. Sting-associated vasculopathy with onset in infancy, or SAVI, is an autoinflammatory disease caused by gain-of-function mutations of TMEM 173.
My name is Lisa Postow, and I'm a program officer in the Division of Lung Diseases at NHLBI, the National Heart, Lung, and Blood Institute. COPD, short for chronic obstructive pulmonary disease, affects millions of Americans. Approximately 16 million people over the age of 18 are diagnosed, and it is estimated that millions more have COPD without even realizing it. Most, but not all people with COPD either smoke or used to smoke, but 25% of COPD cases are due to other factors such as genetics or environmental exposure. Annually, about 150,000 Americans die from the disease. At NHLBI, we are committed to raising awareness of COPD so that people who are at risk for COPD or are in the early stages can get help earlier and get back to doing the things they love. COPD puts a tremendous burden on people with the disease and their loved ones. It can affect quality of life, causing people to not be able to do everyday tasks and stop socializing. It also costs the healthcare system billions of dollars. But not everyone is aware of COPD, and there is a lot about COPD that people don't know. As the name suggests, COPD is a chronic obstruction of the lungs. Lung obstruction means you have trouble breathing. In healthy lungs, airways and air sacs are elastic or stretchy. When breathing in, each air sac fills up with air like a small balloon. When breathing out, the air sacs deflate and the air goes out. For people with COPD, less air flows in and out of the airways because of one or more of the following. Either the airways and air sacs lose their elastic quality, or the walls between many of the air sacs are destroyed, or the walls of the airways become thick and inflamed, or the airways make more mucus than usual, which can clog them. The easiest way to understand how a person with COPD feels is to try breathing through a straw. COPD is sometimes also called emphysema or chronic bronchitis. Both of these obstruct the lungs, but they do it differently. In emphysema, the walls between the air sacs are destroyed, and the air sacs lose their shape. This means the lungs don't work like they should. In chronic bronchitis, the airway lining becomes swollen and inflamed, and a lot of mucus is produced. Most people have a combination of both emphysema and chronic bronchitis. COPD is a slowly progressing disease, and often people don't realize they have it until it's already severe. As the disease gets worse, symptoms usually become more severe, and when symptoms are mild, they may not be noticed right away, and people may adjust their lifestyle to make breathing easier. For example, taking the elevator instead of the stairs, or resting more often. Symptoms of COPD include shortness of breath or breathlessness, excess mucus production, wheezing, and a constant cough, which is sometimes called a smoker's cough. Often people think these are signs of aging or being out of shape. However, extreme shortness of breath is not usually a sign of typical aging.